Hello. Thank you very much for taking the time. My name is Claes Gustafsson, and I am the co-founder here at Atom. I will be talking about navigating infinite sequence function space in proteins. I'll really talk about the tools and the technologies we built in order to use synthetic biology and machine learning to build better enzymes. First, a few words about the company. We were founded back in 2003. At the time, we were called DNA 2.0, and we rebranded the name for various reasons to Atom back in 2016. We're based in the beautiful San Francisco Bay Area. The company is 100 plus people strong. We are completely private, and I've been focusing very much on building technology and building the science over the last few years. So we're very strong on patents. We have a lot of scientific papers published, and we really tried to build a new platform for engineering biology using Silicon Valley-based tools for optimization. So let's talk a little bit about how we see biology. So there are really two dimensions in how we approach this. So one dimension is really the synthetic biology dimension. What are the tools, what are the principles you use to build genes, to build proteins, to build cell lines when it comes to phosphoramidites chemistry, when it comes to cloning, specificity, all these different things, expression hosts, all that. So that's on the x-axis as you build better and better biological entities. On the y-axis is really where we have machine learning to how to design better genes and better proteins and better cell lines, right? So there's no point in making all these different things if you can't design things that are much more efficient than what Mother Nature brought us in the first place. So here in this space, we build synthetic genes, starting with basically phosphoramidites and putting those together and clone that out to get clonal entities. And we can also developed a lot of machine learning tools to design genes that typically express better or have properties that are of value for some reason. The main talk today will be about enzyme engineering, and this is really where you take the synthetic genes and the machine learning to build proteins that fulfill commercial criteria. And how do you optimize, given all the possible options, how do you find an enzyme that fulfills the commercial criteria of your customer? We also do a lot of cell line development. I'm not going to talk about that here, where we build primarily mammalian cell lines that produces various protein pharmaceuticals. But that is where we go into the tools and the processes we are involved in and build. In order to do this, we need to have a nice ecosystem of integrated tools that allow us then to manipulate the biological material in the ways we want. So here I've illustrated, and not going to go through this in detail by no means, but just the point I want to make here is that it really requires quite a few different types of technologies and platforms in order to be able to build and design biology the way we need to do it. So not only do we need to have gene synthesis, as you see here in the bottom right corner, but also we have a lot of the design of experiment, SARs, data integration, and I've really tried to here map it out from the x-axis building axis and from the y-axis design axis, and where all these different tools fit. All the yellow ones here are things where we have built patent estates around. The white ones are tools that we've built a lot of experience and some trade secrets in order to make them work for us. But let's talk about how we do protein engineering or enzyme engineering specifically. So usually we start by going to GenBank. So typically the customer comes to us and they have a starting point enzyme that has some activity or some ideas around what type of activity they need. So we go to GenBank, we go to a number of these big environmental databases. And of course, there are numerous sequences available in these databases. But of course, the total available sequences are infinitely much larger, right? So in principle, you could have any one of 20 amino acids at your end position throughout your protein. And in this infinite space, activity is really very rare. So well, you see, you can look at this as sort of the Pacific Ocean, where you have all this basically just no activity, just water, water, water. And then you have these little tiny specks of islands. You have the Hawaii's over here, you have New Zealand over here, and so on. And those are these little mountains of activity that pops up above this ocean of water. So what we now can use is a lot of the neural net and the pattern matching algorithms to identify what are the properties, what are the patterns that identify an active protein that can sit close by to these Hawaiian Islands versus something that is just that has no activity at all, not folded, has no way of being soluble, so on and so forth. So that gives us then a way to start looking at all the different sequences that are available in GenBank. Now, of course, there's way too many sequences in GenBank to make it even worthwhile exploring every single one. So instead, we built a lot of tools to navigate the existing sequences. And the nice thing with synthetic biology, of course, is you can make anything you want. That's a fantastic opportunity, but it's also constrained by really the cost of expression and cost of assay, because really that's where the big cost, if any project is how do you express these things, how do you purify them, how do you measure the activity. So we built all these tools for sorting through these phylogenetic databases. So here's just some family of some enzyme pulled out of the public domain databases. So what you see here is we sorted these now by color, so from red to green, based off of solubility. And the solubility we identified through both our internal 15 years of protein expression of thousands upon thousands proteins that we built for all sorts of customers over the years and tested for expression. There's also a commercial, an academic, database called ESOL that has similar kind of data. That allows them to very quickly navigate and see how likely this particular protein is to be expressed and soluble in a system. And of course, we have built our own codon optimization algorithm that it really has helped us a lot in order to ensure that we almost always get really good expression levels of all these different things we pull out of the databases. Because at the end of the day, the point is that you want to be able to get as many hits as possible from all the things you pull out of in silico sequences and get as much bang for the buck. So the model what you really try to do here is trying to build prediction models to maximize your bang for the buck. So as we pull out all these sequences, we often test them for all these different properties in order to navigate this multidimensional space that we're typically looking at. We're typically trying to optimize both for activity 1, activity 2, maybe expressibility, maybe TM, all these different things. So here's just an example where we can pull these things out from the phylogenetic tree you saw previous slide, and identify now sequences that are promiscuous, that have activity against in all these different dimensions. Or for instance, in this case, you have this particular sequence that has activity against A, and this one that has activity against A and C as fitness landscapes. And now we can start parsing out that sequence function space in order to better understand how we should. So then what we typically do is we get a starting point. Now we have found something in GenBank or in one of those environmental databases that has properties that are interesting. Certainly has lots of properties that we need to engineer to improve. So what we do is we take the parent sequence, we make what we call infologs, which are homologs that are designed to capture as much sequence information as possible. So in a typical system, we identify substitutions from phylogenetic data. We identify substitutions from structure. And if there is a function available, we certainly take that into account. And when we could sort through all these different substitutions that are available in this family, and we can rank order them based on certain computational premises. And then in a typical set, we take maybe 60 substitutions, and we design, so each of these sequences now on this inflog set will have, say, 3 amino acid substitutions away from the parent. Each sequence is going to be equidistant from any other sequence. And now we design them. So in this case, we're looking at 60 substitutions. So that means we have a 60-dimensional hypercube. We identify 96 orthogonal corners in that hypercube, and then synthesize them. And then the nice thing with these very large data sets being systematically varied is, so in this case, if you have 60 substitutions, that means at 60 positions along your protein, there are 60 residues that can be swapped between wild type and substitution. So the total space you're looking at is 2 to the 60, which is very large. So it says 10 to the 18 here, some number. And this is by only looking at 96 samples on, you have an incredible predictive ability in order to search that very small space using only 96 sequences, very small number of sequences for an enormous space that's 10 to the 18. It really allows you to build beacons of information throughout this. So the way we do it, we do this in Silicon. Now we design these 96 beacons in this 60-dimensional hypercube. We then synthesize them using the synthetic biology. We purify out the protein or what the system now requires. So typically purifying that out of the bacilli or coli or something. And then we test them in a real commercial endpoint assay, something that's high quality, typically low throughput, that allows us to get a lot of information for every single one of those beacons in sequence space. And then based on that information, now we can build machine learning models that allow us then to tell us what substitutions are good, what substitutions are bad for the property, and then we iterate through this. So this is really the matrix that we build around this. So just to walk you through it, so what you have here is the function where each row is a gene variant and each column is now a substitution. So you have residue 1 through some high number, and these can be either wild type or a substitution. So this, of course, is just the top left corner in this really very large matrix. And now you can take each of these sequences now can be measured for all these different properties. So this is really your X data for input data, and then this is your Y data for output data. And now you can identify all these different properties that you care about and build models that try to explain that as best as possible. And before we go into a case study, let's just talk a little bit about what I mean by machine learning, because that's often, it gets confusing at times. So as you get all this data now, you have systematically varied data of typically 96 sequences. And so here I'm going to talk about going back to the matrix in the previous slide, looking at the rows. So these are now each different variant, so typically 96 variants. So how do we model those? Well, what you do is you take out, say, in this case, 15% of the data set, you hide that from the computer. You ask the computer, dear computer, based off of the data that we have, can you make a prediction of the 15% that you don't see? How well are these going to be expressed? What kind of activity will they have in your fitness function? And there will be some predictive value of that. So that's on the y-axis. And then, of course, we measured every single one of these. So we have an X data, which is the measured wet lab data. So for instance, this particular one, let's pick one. So the pink one here, that's the model fit. That's where I have all the data. So this particular one is that fits somewhere. And then the cross val is when you take out 15% and ask the computer to predict it. So for whatever reason, the computer over predicts this one quite a bit, and so on and so forth throughout the data set. But of course, the better the data, the better you have your understanding of your system, the tighter becomes this diagonal as you're navigating towards it. And this now gives you a fit for every single sequence. But not only that, you also get, instead of looking at the now looking at the columns. So what is the contribution for each of the columns in your matrix? So what you're looking at here now is the same data, but now instead looking what is the contribution for each of these amino acid substitutions. So for instance, going through from left to right here, you see that some substitutions are most of them are around 0. They're fairly neutral. There are a couple of guys that are horrible. So this variable 42, for instance, is very, very bad. Variable 48, for whatever reason, is a really good one, has very positive activity. And then you see that the distribution bar is really a measure of the epistatic effect, the context effect of each substitution. Because as we know, biology does amino acid substitution never works alone. They always work in the context of the enzyme itself. So what you see here now is that the error bar really is an indirect reflection of how epistatic or how context dependent that particular residue is. So for instance, this one is variable 17 is for whatever reason fairly context dependent, whereas this one up here it's nice and tight, so it's very linearly additive. And that allows you now to get measures for how you navigate this space and how to optimize it for different functions. So I'm going to do a case study. In this particular case, we do a lot of work nowadays with antibodies. So I'm just going to start with a commercial antibody, the commercial molecule we've been working with. And in this case, we identified 41 residues in the heavy chain where we can introduce substitution. So that means that at 41 residues, you can either have the wild type amino acid substitution or the wild type amino acid or the substitution. So total space in the heavy chain is 2 to the 41. Two options at 41 residues. Space is around 10 to the 12. In the light chain, similarly, we identified 36 residues where we can identify amino acid substitutions that are worthwhile trying for a swap. And so in this case, we ended up with 2 to the 36, which is 10 to the 10. And actually, if you put these two together, the proper math on it ends up being a total space of around 10 to the 23. So it's a fairly significant space that we're trying to explore. And here we're starting with a commercial molecule. So we're starting with something that's already on the market, that's already been battle tested, that's already presumably been optimized at some context for some customer in a previous existence. So this is what we call a matrix slide. So here we'll look at how we navigate through this multiple amino acid substitutions. So each column here is one amino, and in this particular case, one antibody. So, for instance, antibody number 1, the very first one, this blue set of 96, has no substitutions at all in the light chain. So these are all light chain mutations, and these are all the heavy. So the very first one has no substitutions in the light chain, has 3 substitutions in the heavy chain, this one and this one and this one. Similarly, this particular substitution is seen in antibody 1, antibody 4 maybe, antibody 10, and so on and so forth across this data set. So in the blue round, half the data set we only do substitutions in the heavy, and half we only do substitutions in the light. And then we synthesize these and we express them and we purify them, and then we test them. In this case, we're looking at primarily for binding. So what you see here in the subsequent, the pink round, is we identified a number of substitutions that were really poor. So for instance, all these substitutions that are between this part of the molecule were all worse than the starting point. So we took those out and never going to use them again. And contrary is, for instance, this substitution right here, it turns out to be really good when we do the modeling. So we freeze that into the backbone and keep that for all the possible substitutions, all the possible antibodies going forward. So this allows us now to very rapidly narrow down the space. So you see the amazing thing here is in blue round, we're looking at a total space of 10 to the 23, which of course is an immense number. And only by doing this very rapidly, we can funnel down all this sequence function information into a very small data set that is just 10 to the 4. So massive reduction in sequence function space, and allow us then to very quickly navigate for improvement in this data. So what you see here is that in the light chain, a number of the sequences are frozen in. There's still a couple of uncertainties here, but they're much more consistent, either good or bad, versus in the heavy, there's still a lot of unknowns. We can take this information now, and then we can look at the sequences themselves and measure binding. So what you're looking at here in the blue round, binding comparing to the starting point parent, Most of them are worse than the parent. There are a couple of guys that are a little bit better, and you have a nice distribution. That means you can really build models that allow you to put weights on all these different substitutions. And I really want to point out, you going from the blue round to the pink round, how you not only get a couple of hits, but you really move the entire population that you're screening for towards a much higher binding. And so here we're looking at Ka, which is 1 over Kd for antibody enthusiasts. And based off of this information now, we're going to 10 to the 4. We can then start cherry picking a few guys that we do here in the green round to allow us then to navigate this even better. So, but of course, it's not only binding that's important, but really it's titer. In this case, the titer and TM was also critical. So here we're looking at now in sets of 3, so 3 bars for each amino acid substitution, how well each substitution contributes to TM in yellow. Pink is titer and binding is blue. So what you see here is just as you go through this, now you can look at each substitution by itself and see how well it contributes in all these different dimensions. And it really becomes more or less a spreadsheet decision how you move this forward. It's a really powerful way to very rapidly navigate very large and complex spaces. So now we can take it and also look at these sequences and how this is the same data plotted instead as sort of a 3-dimensional where you have binding on the x-axis, you have titer on the y-axis, and you have TM as color. So what you really want is something in this top right corner up here that is binding really well, has a good titer, and is as red as possible in order to have a good TM. And so what you see here is the parent, and these are now 3 sequences that pulled out. So so you can basically follow sort of a Pareto front going towards this optimization. And now you can pull these particular numbers out, and you can really have 3 different options on or as many as you want for sequences that fulfill your commercial criteria. So that's for 3 different fitness functions. But of course, as we go through this, we get more and more complicated product, and we get many different fitness functions that all needs to be optimized for in context. So here's another example. This is an enzyme we did for a commercial partner, where we're looking at 8 different fitness functions. So we're looking at the expression, solubility, and there's a functional assay. In this case, it had an on rate and an off rate for binding, low pH, high pH for various reasons, and of course stability. And we measured this now. So now we're looking at variables, all these different variables. A variable here could be an amino acid substitution, but of course it can also be pairs of amino acids, or it could be anything you want it to be. And what we can now identify and put weights on each of these variables for 8 different fitness functions. And of course, now it gets really complicated. You have a high degree of complexity for every single one of those substitutions. And some are better and some are worse. And how do you navigate when you're looking at 8-dimensional fitness landscape? So in this case, we talked to the customer and really try to understand not only how do you optimize for all the 8, but how do you as an end user value each of these fitness functions? So it turns out that for this particular customer, fitness function A was the commercially most relevant fitness function. So they say that the value of this one is approximately 6-fold over fitness function B. So now we can put different weights for all these different fitness functions, and then combine them just by additivity, and then build a model where we try to predict the relative contribution for all these substitutions now in this integrated fitness function, summarized fitness function. And so what you see here now, variable 1 through 5 are more or less neutral, slightly positive for this combined fitness function. Whereas, for instance, this variable over here is just horrible, and this one is really good, and so on. And now you can actually take this model and you can build a model for all the X data, for all the sequences, and look at the cross now between calculated fitness and predicted fitness, with calculated being from the wet lab, combined from all the data. And what you see is you have a surprising degree of correlation. This is I love this slide. This is a fantastic slide. really allow you then to build models that combines all these different fitness functions into one unifying algorithm. And so in this case, for 8 different fitness functions, our current best is we did for a customer, I think 258 fitness functions all combined into one for a commercial application. And of course, at the end of the day, as in an antibody, it's not just binding, it's not just a KCAT over KM for an enzyme, but it really is a number of these different fitness properties that you try to optimize for, all in parallel to make sure that you are reaching a commercially relevant molecule. And so we actually just the other day, we sat down and tried to enumerate all the different types of enzymes we've been working on. And I think we end up in the 40 to 50 or so different projects. And there must have been at least 30 to 40 different classes of enzymes in that data set. So we've certainly built a lot of experience and a lot of knowledge over the years and doing this for a number of companies, small and large, everything from very large corporations to small startups and everything in between. And that allows us then to really utilize the efficiency of biology, because at the end of the day, biology is just ACGT. It doesn't really matter if you want to make a renin for cheese production or you want to make an antibody to cure cancer. At the core of the algorithm, it's all completely redundant algorithm-wise. So you can really overlay all these different things on top of each other and capture the sequence function data. Because you're looking at, for an enzyme engineering, you're looking at things like KCAT over KM, but also you're looking at maybe stability, temperature stability. You're probably looking at expression yield, protease stability, activity in different pHs, and all these different parameters. And it's going to be derived specifically from the customer's knowledge and domain expertise in order to make that protein a commercial success, in order to really drive that to market. So that makes us really, really excited. And I think that's the opportunity that really opens up here in life sciences as we're getting closer and closer to really replacing petrochemical industry with biology. And with that, I just want to thank you all for spending time with me. I appreciate it. If you have any questions, feel free to ask that at the Q&A, or you can reach me at that email address you see below, cgustafson@adam.bio. And thank you very much.